I'd like to thank the Colorado Bioscience Association for inviting me here to speak today. Actually, I first heard about this initiative when I was a graduate student at Purdue, and it was one of the things that drew me to Colorado and to CSU. So that's a good note there. Today I'm here to talk to you about Phyto Detectors. I'm a postdoc in Dr. June Medford's lab in the CSU Biology Department. And we are interested in using plants to detect environmental pollutants. Recently, a 2007 study by Mount Sinai, Johns Hopkins, Harvard, and the Smithsonian Institute identified those pollution sources which most negatively affect human health. They paid particular attention to those pollutants which most negatively hurt children. They came up with mining, industrial pollution, Cold War era, military pollution, and energy companies, gas and oil, were the sources for this pollution. Here in the U.S. alone, the cleanup costs estimated with cleaning up toxic heavy metal pollution is $350 billion, and that was in 2000, that was estimated. With places like Leadville, Colorado in the news on CNN, you can see why. In addition to heavy metals, organic pollutants such as MTBE, which was added to gasoline as an octane booster, has leaked into our drinking water supplies, aquifers, and is carcinogenic. The maps shown here are the areas in our country with MTBE pollution. I drew a black circle around our area, so hopefully this message will hit home. While plants have been used for environmental remediation, a process called phytoremediation, which unfortunately is underutilized, not only in this country, but also in the state. So plants can extract, degrade, and clean up pollution, and this is published. Little was known, however, how plants might be used for environmental detection. What do I mean? Well, phytodetection. So plants as specific detectors or sensors. So we wanted to investigate this. Luckily for us, plants have evolved over 420 million years to sense and respond to required changes in their environment. Substances in the air, such as ozone, ethylene, are easily sensed by plants and often result in a response. Chemicals and elements in the soil are also detectable by plants, such as selenium, another big problem we have here in Colorado. And this sense and response is very nonspecific, meaning plants go through multiple signal cascades in response to a given environmental cue. So we wanted to ask the question, can we make plants which sense and respond after coming into contact with a specific toxic pollutant? For example, a canary in a coal mine, except for we don't want to kill our plants. So specific detector plants would need two traits. The first trait they need is the ability to sense something specifically of interest, a sensing system. The second thing is they need the ability to respond in a way that humans can see, so a readout system. And that's what we're going to talk about first, is our readout system. So a good readout system would have three requirements. First, it would be quick. Second, we'd be able to look at this readout system and detect it remotely. Thirdly, we'd like to be able to have the public, the general public, recognize our readout system. So we started thinking, and looking here at the leaves changing colors here in Maine, we noticed that the evergreens are easily distinguishable from the plants that are the the deciduous plants that are actually changing color here. And so the green pigment chlorophyll, or the loss of chlorophyll, is a potential readout system. So in order to do this, we needed some way of genetically manipulating the plants to rapidly, in this case, degreen. And so what we did was we needed a simple means to link chlorophyll levels to a specific input. So we designed a synthetic gene circuit, or a degreening circuit. And what we did was we simultaneously stopped the synthesis of chlorophyll and initiated its breakdown. So this would happen very quickly. In the environment, it could take a week for a plant to go from green to yellow, and then for its leaves, or even longer, to fall off. So this is our functional plant detector, and here we are sensing the explosive TNT, which often leaks out from inside old landmines. So TNT is activating our degreening circuit. And what you can see here in the laboratory plants, tobacco and Arabidopsis, is that after we add TNT, this is right after the addition at 0 hours, but after 1 day, we're starting to see a decrease in the chlorophyll. After 2 days, even less. After 3 days, even more. And when you compare this degreening over time to our controls that do not have this circuit, you can see that these do not degreen, whereas in the laboratory plant Arabidopsis, over 3 days, we see this degreening from the start. So we see this induction with only 23 parts per trillion TNT. That's the amount of 5 drops in 2 Olympic-sized swimming pools. So it's very high detection limit. So we also wanted to hopefully be able to detect these things in the soil. And sure enough, what we're looking at here is root detection in the soil of TNT. So we add the TNT to the soil, and in our Phyto Detectors, that's shown here, we see this degreening over time. This is compared to our wild-type control plants treated with the same TNT. We don't see this. Now, one thing we know is that chlorophyll fluorescence can be easily measured by machines. And so what this is showing here is the chlorophyll fluorescence spectra across the top here as these plants degreen. And so as you can see out here at the end, after 96 hours, our most degreened plant shows quite a different chlorophyll fluorescence spectra than the control plants after 96 hours. Well, this is important because we also like to be able to detect these things using machines and inexpensive handheld devices. So using a handheld device here, we can detect changes in chlorophyll fluorescence in 2 hours. Using satellites, we can detect changes in chlorophyll fluorescence in 3 hours. And so this is very important for remote detection. So limitation of all previous biodetectors is that you only get one use. Phyto detectors reset or regreen once the TNT is removed or is absent. So here we're showing plants that have degreened with TNT, and after 3 days of growing these plants on media without TNT, they regreen. So our process can be repeated multiple times. Well, how do we sense specific substances? Well, we used computers to design a protein receptor that is highly specific. And this provides us the ability to make a sensor to a substance of interest that won't sense other things. And so our protein receptors are made to fit or bind to a given pollutant precisely. So the functional plant detector consists of the following. The computer-designed chemical receptors for, in this case, TNT. This receptor is targeted to the surface or outside of the plant, which then talks to our synthetic signal transduction system, which then talks to our readout system, which is our degreening circuit. So plants used in detector technology, in this technology, are extremely specific. And our receptors can be designed to most all small pollutants, including most of those on the EPA's website. So the question is, why use plants? Well, plants are already in the human world, and they're in important places, like the White House, in offices, in airports, in shopping malls, around cities, in polluted industrial areas, and around mining sites. So our gene circuits can be placed in plants to allow inexpensive and widespread detection of pollutants. For example, around pipelines where you might get leaks or explosives. And we hope that this technology will empower ordinary people to detect pollution. In our lab currently, we have detector plants for TNT, the high explosive in landmines, also for MTBE, the carcinogenic gas additive, and we're working on detector plants for toxic heavy metals such as lead, arsenic, and mercury. The vision of our products is that phytodetectors will enhance environmental detection around pipelines. For example, you see the plants here, and if a leak were to spring, these plants here would turn white, and so this is easily detectable. Environmental pollutants we don't know might be out there in certain areas, waterways, around other manufacturing areas, in landfills. And when we want to first detect before we clean up these areas, with phytodetectors, we can do that. And so you can see this is a mock diagram of how this might happen. You can see it's in here, and then there's where the white color comes in, and then here and here. Based on our findings in the lab, Dr. June Medford and CSURF have now started Phyto Detectors, Inc., and have adopted the slogan, "Environmental detection coming to a plant near you." With that, I'd like to give special thanks to Dr. June Medford, Dr. Kevin Morey, Dr. Mauricio Túnez, Tessa Bowen, Kirk Albrecht, Jeff Zudnik, and our funding agencies, Office of Naval Research and the Colorado Bioscience Association. Any questions?